Swayam Prabha. Digital India. Educated India.
Welcome to the next session on Organic Chemistry Paper 3. In Unit 5, we are studying about terpenes. In Lecture 1, we are going to study about terpenes, occurrence and isoprene rule. I am Dr. Balaji, currently working as Associate Professor in the School of Biotechnology at Jawaharlal Nehru University. This project is sponsored by DTH Swayamprabha, MHRD, New Delhi. Let us look at the topics that will be covered in this session. We are going to look at the occurrence of terpenes. We are going to study about the natural occurrence, where they are present in the plants and flowers, and how they are present as in the case of herbs and spices, and in the plants with the medicinal uses. So what are all the different places these terpenes are actually present? We are going to look at various examples, and then we are actually going to study about the isoprene rule, which is the basic for terpene chemistry. We are going to look at what is an isoprene rule and how it was developed. We are going to see, and finally we are going to study about the gem-Dialkyl rule also. Welcome to the session on terpenes. We are going to study about occurrence and isoprene rule. Let us look at the brief overview of terpenes. The name terpene, or in German it is called as Terpen, was suggested by Kekulé for the compounds having C10 and H16 molecular formula hydrocarbons. Basically, this is a monoterpene. We will study about the molecular formula later, but the name terpene is actually suggested by Kekulé for compounds having a general formula C10H16. These are terpenes. When we say, these are basically hydrocarbon compounds, and when we say terpenoids, the terpenoids have oxygen containing terpenes. That means that oid, when we add as an extra word, we mean to say that the particular compound contains oxygen. And in fact, the terpene is the largest class of compounds with more than 80,000 compounds so far identified. So this is the major or the largest class of compounds. And all the terpenes end with the name ENE. That means all hydrocarbon terpenes will end with the name ENE. And terpenoids may have different names, but terpenes, since they are hydrocarbons, they end with the ending ENE, and these are basically volatile compounds, and they evaporate very quickly, and they also undergo oxidation very readily to produce alcohols and other products. Isoprene, basically a gas, is the second most abundant volatile substance that is present in atmosphere after methane. Methane is the natural gas which is abundantly present in the atmosphere. After methane, isoprene is the gaseous molecule which is present in the maximum quantity, and this is also a volatile substance. So isoprene is basically the terpene molecule. The monomer unit of all terpenes starts from isoprene. And these terpenes are different types of compounds, and they function as defense molecules in certain plants. They are also involved in photosynthetic pigments or as phytohormones. Like that, they function as different way in different types of plants. For example, in some plants, they attract pollinators. That means they behave like an enabler. Whereas in some other plants, they function as a predator to repel the predators. So that functions as a defense mechanism. So in some plants the terpenes behaves like an enablers, and in some other plants they behave like a defense person. So that is the different role played by terpenes in the plant kingdom. And these are also involved in various other functions, like the components of electron transfer systems, or protein modification agents, or even in membrane fluidity determinants and as antioxidants. So these are all the various roles played by terpenes. And these terpenes are widely found in flavors, fragrances, spices, perfumery, and cosmetic materials. So we will look at what are all the different types of terpenes present in various aroma plants we will be seeing right now. And in fact, why the terpene is very important is actually can be understood by the Nobel Prizes given in chemistry to this particular field. Otto Walsh, who is considered to be the father of terpene chemistry, received the Nobel Prize in 1910. So he identified the structure of various terpenes, and in fact we will be studying about some of the work which was carried out by Walsh in the structural determination of a few of the terpene compounds in the later part of this course. And he is considered as the father of terpene chemistry, and he meticulously identified the structure of various terpenes. And Richard Kuhn and Lippard Ruzicka are the two other people whose contribution to terpene chemistry is enormous. So these people specifically received the Nobel Prize for their work towards terpene chemistry. And other people who are shown here, like Will Starter, Wieland, Windus, Fischer, Hans Fischer, Paul Carrer, or even John Confroth, Vladimir Pralog, E.J. Corey, R.B. Woodward, Robert Robinson, and George Vola, are all these people contributed to some part of terpene chemistry. For example, R.B. Woodward had synthesized various vitamins, and Robinson also had identified the structures and synthesized various terpenes. E.J. Corey also contributed to prostaglandin, steroids, and other compounds synthesis. George Vola worked on carbocations, especially alpha terpineol and other type of carbocations, how they undergo reactions. Some of these things he had also studied. So basically there are various people who had contributed to the development of terpene chemistry, and this clearly tells the importance of terpenes in chemistry. And let us look at the occurrences of terpenes. Terpenes are basically plant secondary metabolites, so this is the most important thing we have to remember, and they are mainly found in aroma plants and essential oils. So this is the place where the terpenes are actually found, and they have different molecular size, and they have very high structural diversity. When we look at the synthesis biosynthetic pathways, we will understand how a single isoprene molecule can be modified or converted biosynthetically to different types of skeletons, we will see. So the terpenoids are having a different molecular size, and they have very high structural diversity. And the oxygen functional groups, when we talk about in the case of terpenoids, the various oxygen functionalities are alcohol. Say for example, if you look at menthol or nerol, they have the hydroxy unit. When we talk about an aldehyde function, citral and nerol are having an aldehyde function. And when we look at camphor, the camphor contains ketone moiety, and citronelyl esters contain an ester moiety, and eucalyptol contains ether moiety, and thymol, a good medicine for a cold, contains a phenolic unit. So these are all the various oxygen substituents that are present in terpenoids. And most of the terpenes are having antiseptic properties, and they are actually insoluble in water. So let us look at some of the natural occurrences of terpenes. So the term or the Greek name for terpene was actually derived from the terebinth tree or the turpentine tree. So this is the tree that is shown. From this tree only the turpentine was first isolated. From the turpentine, terpenes were found later on, and that is the reason the name for these kind of compounds were given based on the Greek name of this particular tree, that is terebinth tree. Amber, if you look at the fossilized materials, especially the polymerized or the fossilized materials which came out of the plant. In fact, if some of you had seen the movie Jurassic Park, so there they talk about some of these kind of fossilized compounds which are present. So that means these are very old materials that is present, and this particular the whole material, the orange color materials what we are seeing here, is basically called as an amber, and this amber is basically a terpene moiety, a polymerized terpene moiety. And of course, we have various type of terpene scents. For example, in Indian houses, many of the people use agarbatti, dhoop, cone stick, sambrani, and etc. So some of the things are actually given here in different forms, and all of these contain terpene moieties. And in fact, pitch is another terpene compound which is generally used as an adhesive to waterproof boats and ships. So here you can see this particular is the pitch, and this is used as an adhesive to connect two things. Like this is basically a rock and this is the wood, so the wood and rock are connected through this pitch. So this is another type of terpene compound. And rubber, as all of we are using it on a regular basis, is basically a natural terpene polymer, and this is actually coming out as a white liquid from a plant. The rubber tree is giving this as a white liquid, and this is where the rubber is actually present. So naturally many of these terpene occur from the plant. And if you look at the chemicals that are present in these kind of compounds, say for example when we look at the beta carotene is actually the source for vitamin for all humans, so it is having the structure as shown here. And another terpene, lycopene, is actually the component of carrot and watermelon. This gives the red color. So here is the compound lycopene. This gives a red color to these kind of compounds, and orange color of this carrot is also due to some of these carotenoid compounds. We will look at some of the carotenoids compound later. And the ginger, whatever we consume, contains a gingiberine molecule, and the gingiberine structure is actually shown here. Taxol is used as an anti-cancer drug. This also has a diterpene. So here we see the terpene unit that is present here. This is a tricyclic system. And vinblastine, artemisinin are some of the drugs which are used to prevent either cancer or malaria. The vinblastine structure is shown here, and the artemisinin structure is shown here. So these compounds all contain, basically these are terpene derivatives. And when we look at the flowers and the plants, especially the aroma flowers and plants, when we look at the orange peels actually contain limonene and cymene. So you see the typical smell of the orange peel. So that smell of the orange peel actually comes from these kind of terpenes. And when we talk about the wine grapes, this contains actually geraniol, a terpenoid. And carrots, the color of this carrot comes from cymenes. And wood chips, when we look at the pine smell of these wood chips actually comes from pinenes. And cannabis actually contains myrcene as another terpene. And if you look at lavender, the beautiful lavender colors, lavender smell comes from the linalool. And the lemongrass actually contains nerol, which gives a typical that lemon smell to this particular compound. So various terpenes give different aromas to the particular plants. In addition to that, there are various herbs and spices which contains terpenes. Here is a big list of compounds which is generally used in our regular customary Indian food. Especially many of the masala items what we are consuming are basically terpene compounds. Say for example, the basil leaf or the coriander leaf which we are using contains couple of terpene compounds or the terpenoids. So the basil contains eugenol, camphor, limonene are some of the terpenes that are present in basil, and borneol and geraniol are present in coriander. So this gives a different flavor to our food. And when we look at bay leaf, cardamom, cinnamon, clove, fennel, all these things contains different terpenes. For example, the bay leaf what we use for our masalas, especially for making biryani and pulav and other things, contains eugenol. And cardamom contains nerolidol and alpha terpineol. Cinnamon contains eucalyptols and linalool. Clove contains eugenol. Fennel contains fenchal, linalool and other compounds. And in fact, the garlic contains geraniol and the black pepper contains limonene. So these are all the various terpenes that are present in the herbs and the spices which we consume on a regular basis. So these compounds actually give the flavor to the food what we are eating. So the terpenes are very, very essential compounds for our consumption. Now let us look at some of the medicinal uses given by these kind of terpenes. These terpenes are generally present in various plants. So let us look at some of the compounds. Menthol is generally used for the cold. This is actually the structure of menthol is shown here, and this is basically a terpenoid compound. And when we talk about borneol, this is used to treat sleeplessness, and this again is a terpenoid compound. Linalool was used as a sedative and antidepressant. So the structure of linalool is actually given here. And ingenol 3 mebutate is generally used for actinic keratosis. So the structure of the compound is actually given here. And we also have other compounds like tapsigargin, farnesol. Farnesol is used as an to reduce the allergic which we get from asthma, edema and gliosis. The structure of farnesol is actually shown here, and the thymol is generally used for common cold and other things basically, and the structure of thymol is actually given here in this structure. As we mentioned earlier, this is having a phenolic unit. So these are all the various terpenoid compounds which are generally used, or they are generally used for the medicinal purpose. And of course there are other compounds also there that what we have seen, Taxol and artemisinin, vinblastine. All these compounds are also examples of terpenoid compounds. Let us look at briefly the biosynthetic pathway, how terpenes are actually made. So all terpenes are derived from the C5 building block, and this C5 building block can be isopentenyl diphosphate, as the structure is shown here. So we have the 2-phosphate unit, and another compound may be dimethylyl diphosphate or DMA-DP. So this also has a 2-phosphate units, and this is the C5 structure we are talking about. So we have 1, 2, 3, 4, 5. So total 5 carbons are there. Similarly, in the case of isopentenyl unit also we have, let us start from here, 1, 2, 3, 4 and 5. So this is not the IUPAC nomenclature which I am actually using here. I am just numbering the carbon atoms. So we have 5 carbon units. So that means all the terpenoid compounds are derived from the basic C5 unit, and this IDP or IPP and DMA-DP are the basic starting points, are the monomer for all the terpenes. And these terpenes are basically produced by 2 different routes. Biosynthetically they follow 2 different routes. One is called the mevalonate pathway or MVA pathway, and the second one is called 2C-methyl D-erythritol 4-phosphate or the MEP pathway. So all the terpenes are generally produced by either one of these pathways. So we have been talking about the C5 unit. So the C5 unit is actually called as the Isoprene unit, and Michael Faraday in 1866 first observed that isopentene from rubber. So he first observed that natural rubber actually contains a compound isopentene. So this happened in 1826. So he was the first one to find out that there is a C5 unit present in rubber. And then in 1860, the Williams actually isolated this isopentene from rubber. So first Faraday found out the presence of isopentene in rubber, and William actually isolated isopentene from rubber. So this again, isopentene is nothing but a C5 unit. So this C5 unit is a basic unit that is present in a polymer called rubber. Then in 1866, Kekulé gave the name terpene because he found out the turpentine oil contains some C5 units. So he gave the name terpene to these kind of compounds, and that is how the first name was given to terpenes. And in 1884, Tilden first isolated the turpentine oil, that means isoprene he obtained from the turpentine oil, and this isoprene dimerized into dipentene. So that also he had found. He dimerized this by two different methods. One is by acid treatment, he dimerized the isoprene into dipentene. He also used heat to convert the isoprene into dipentene. So either acid treatment or heat produced the dipentene from isoprene. So this is the next major development in the field of terpenes. And then in 1897, finally the isoprene structure was established. So what is the structure of isoprene was established in 1897 by Ipatu and Wittoff. So this is how the relationship between isoprene and terpene are slowly evolved over a period of 60 to 70 years. And now we are moving on to the next important thing, that is called the isoprene rule. As we saw, all the terpenes contain a C5 unit. So the C5 unit is nothing but the isoprene unit. And the Ruzicka, who received the Nobel Prize in 1939 for his work on terpenes, first proved that something called the isoprene rule. And what he showed was, through the biogenesis, all the terpenic compounds were formed from the simple isoprene rule or from the isoprene unit. So this isoprene unit is basically the monomer which was responsible for the formation of different types of terpene, and he actually formulated this isoprene rule. And for this and other various work related to terpene, Leopold Ruzicka received the Nobel Prize in 1939 in chemistry. So he formulated that the terpenoids are actually formed by the enzymatic reaction. So he talked about the biogenesis or the biosynthetic way by which an organic compound is actually made, and he talked about the enzymatic cyclization of the substrate alkene chain. So that means isoprene has two alkene unit. So these two alkene units actually combine in different ways to produce different types of terpenic compounds. So they may be condensed most of the cases in a linear fashion, that means head to tail fashion, and the condensation of various C5 unit leads to the formation of the monomer, dimer, trimer, polymer, etc. So that is how all the different types of terpenes are formed. So here we have three different types of combination. So always when we are numbering, we always start with the branch getting the lowest number. So this is called the second carbon. And always remember, whenever we talk about the isoprene rule, what is head and what is tail is confirmed by the presence of the methyl group. So the methyl group is closer to the head. So this is the only thing you have to remember when we talk about isoprene rule. And there is also, because we have two double bonds, and out of the two double bonds we are giving the lowest number to this methyl unit, and this is this side is actually called as the head unit, and the other end or the other alkene is the tail unit. So head, H, when we say H means it is head, and T when we say T is tail. So the head to tail combination actually leads to the different types of terpenes. So sometimes the combination may be head to head or tail to tail or head to tail. So all the three combinations are possible. The most preferred one is the combination of head to tail combination. So this is where when we have the methyl group at this particular alkene, nearest alkene, so this is called as the head. And when the methyl group is far away from the alkene unit, so that is what is called as the tail. So the head and the tail combine together to give a dimeric unit. And there is another way of combination where two heads can be combined together. So this is one way of connecting. And there is also a third way where both tails can be connected. So these are all the three different ways by which the isoprene unit can be combined, and this leads to various type of terpene and terpenoid compounds. So that is why this rule, that is called as the isoprene rule, is the most important rule in the study of terpenes. And let us look at some of the examples of this isoprene rule. We have couple of compounds given here. This is a linear or a acyclic one. This is a monocyclic one. This is a bicyclic one. So we are actually going to look at three different types of terpenes. So in the case of linear or the acyclic one, we are going to look at how the isoprene rule is actually applied. So myrcene is one of a compound. So here we have a unit starting from, this is the carbon number 1. For convenience, I had just put the number as here. So we can assume that this is the head and this is the tail. So wherever it ends is the tail. And as I mentioned, whenever we have the methyl group, so that methyl group carbon is considered to be the closest to the head. And similarly, we have a methyl unit, or here that alkene unit is there. So this can be considered as the starting point. So when we say this one, so we call this as the head unit and this is the tail unit. So we have combination. A head is connected to a tail. So this is what is the combination found in myrcene. And when we look at the geraniol, here again there is one head which is connected to another tail. So we can actually number them accordingly to find out which is the head and which is the tail. So always remember, whenever we have the methyl unit, that is close to the head, and that is why here we have a methyl unit and this is close to the head, and the other end is called the tail. So here we have this methyl unit, so that is why this geraniol, this is considered as the head and this is other end is the tail. So head to tail combination lead to the formation of an isoprene unit. And when we talk about the monocyclic systems, here again we have a head unit here and there is a tail unit here from one part, and here is the head unit at the bottom and here is the tail unit that is present here. So now this head and this tail are actually combined together, and the middle carbon and one of the tail is connected together to form the cyclic system. So this is what we call like a head to tail combination. So alpha terpene is formed by the head to tail combination of an isoprene unit. Similarly, carbone is also formed, as here we can see these are all the two carbon atoms between which the bonding is formed. So here again it is a head to tail formation. Of course, other functional groups are introduced later on the biosynthetic route, but we are mainly focusing on the carbon unit attachment only according to the isoprene rule. Similarly, when we go into the bicyclic systems, we have alpha pinene, beta pinene and camphene. All of them have different types of rings. Say for example, here we have a 4-membered ring and a 6-membered ring. Here we have a 6-membered ring and a 4-membered ring. So these two are basically formed by the combination of two isoprene unit, and we are going to see there is one head here, there is one tail here. So these are all the ways by which these two are actually connected. And in many of these kind of compounds, at least one cyclization or one point of attachment is sufficient to be used for the isoprene rule. Of course, when you look at the second connection, we cannot formulate a head and tail connection in the second one. That is the reason some of these compounds are actually formed by the biosynthetic route and various further modifications. So for defining the isoprene rule, we are mainly focusing on at least one attachment that is responsible based on this isoprene rule. So alpha pinene has 1 and 4, that means head and tail combination here. Now when we look at the beta pinene, this is also having a 1 and 4 carbons are actually connected through this isoprene rule. So we say this again is a head to tail combination. And when we look at camphene, we are looking at here, that is 1 and 4 carbons which are connected to each other. So this is what we call like a formed by the isoprene rule. So this way we can actually use isoprene rule to find out how the different types of C5 units are connected in these kind of terpene compounds. And here some other terpenoid derivatives are given. So some of the terpenoid means they actually have an oxygen unit that is present in here. So when we talk about farnesol, this has a hydroxy unit. When we talk about retinol, this is also having a hydroxy unit. And nepeta-lactone has a ester unit, that is the internal ester, that is the lactone is present here. So if we look at all these compounds, here again we can actually use the isoprene rule to find out how these combinations or the connections would have happened. So here is the head and tail connection that is shown here. And in the case of farnesol, we have one place here is a head and tail combination. In another place, we have another head and tail combination. Similarly, in the case of retinol, within the cyclic system, we have a head and tail combination. And we also have a double bond, which is also formed by the head and tail combinations in two places. So there are further modifications which led to the introduction of the double bond. But basically, when we talk about the biosynthetic formation using isoprene rule, we can simply look at the connection points only. And similarly, we also have other bigger systems where in the case of abatic acid, we have multiple isoprene units are actually connected together. So here we have the head and the tail combination. And here again we have the head and the tail combination, but in the other case we have actually tail to tail combination. So there are three different combinations that are possible in this particular case, and that leads to the formation of abatic acid. And of course we can exactly say there are this is one isoprene unit, and this is a second isoprene unit, this is a third isoprene unit, and this is a fourth isoprene unit. So there are four isoprene unit which are combined together to give abatic acid. And when we look at humulene, here we have the head and tail. If you look at here, this is the head and this is the tail. So this is one combination. Here again we have the head and the tail combination. So, and the next one is here only the tail to the other carbon is actually attached here. So we have three, if you look at this is one isoprene unit shown in blue color. There is another isoprene unit shown in red color. There is the third isoprene unit shown in the green color. So we have three isoprene unit combined together to give humulene. So based on that, we can actually break every terpene into simple isoprene unit using this isoprene rule. And let us look at some other bigger structures also. For example, if you look at squalene, squalene has actually, this is the one isoprene unit, this is the second isoprene unit, we have the third isoprene unit, here we have the fourth isoprene, here we have fifth, and here we have sixth. So total 6 isoprene units are actually combined together to give squalene. But one unique nature of the squalene is basically we have a tail to tail connection. So this is a very crucial or very unique thing that is present in the case of squalenes. And we will be looking at how these kind of tail to tail connections are actually happening through the biosynthetic pathway. We will look at in the later part. But one thing you have to remember is even squalene, which is a pretty long molecule, has total 6 isoprene unit connected to it. So most of, or if not all, the terpenes can be broken down into the simple isoprene unit. And let us look at lanosterol. This is basically an example of a sterol, and this sterol is again it is a terpenoid compound, and we are going to look at what are the different isoprene units that are used for the formation of lanosterol. So here we look at the structure. So here this is one type of bond. So the dotted lines are showing what are all the different places where this connection is actually made. So from this connection we are actually going to assume that if these points are the place where it is actually dissected, then how many different units of isoprene we are going to get, we are going to actually see here. So if I cleave this place, so we are actually going to end up with 1, 2, 3, 4, 5 and 6. So 6 different isoprene units combined together to give the lanosterol molecule. So this is how we can actually use isoprene rule to find out how many isoprene moieties are present in any complex terpene. So we talked about the natural rubber, and the gutta percha is another type of polymeric material. So here again we follow the same isoprene rule. So if you look at the structure here, we have the connection here. In every place, the one which is written in the black color is the place of connection between two isoprene units. So if you look at the number of carbons here, we have 1, 2, 3, 4 and 5. So every 5 carbon unit or the isoprene is connected towards the cis combination. So in the case of natural rubber we have the cis connection, and in the case of gutta percha we have the trans connection. So that is the only difference between the natural rubber and the gutta percha. Here again we have multiple isoprene unit that are connected. So this is an example of a polymeric terpene, and both these compounds also follow the isoprene rule. And if you look at the Faraday, so he obtained or he identified the presence of isopentene from the natural rubber through this particular combination only. And let us look at the gem-Dialkyl rule. So this is another important rule in the case of terpenes. When we talk about the open chain monoterpenes and the sesquiterpenes, they actually undergo cyclization to give cyclic systems, that is a cyclohexyl ring. And when they have this cyclohexyl ring, basically there is something called the gem-Dialkyl unit, because when we talked about the isoprene unit, so we wrote the structure like this. There is a double bond, there is another double bond. So that means we have one methyl unit here and another CH2 unit which may be converted into a methyl unit later on. So we actually have a gem-dimethyl group that is present in all the terpenes. So these gem-dimethyl, if they are present within that cyclic system, that is going to make that ring unstable. In other words, the gem-Dialkyl group or the gem-dimethyl groups render this 6-membered ring and the semicyclic double bond highly unstable. So that means these kind of gem-dimethyl group cannot exist within the 6-membered ring, but they are actually kept outside of the 6-membered ring. So in the cyclic system they are not present because they lead to instability in the case of simple 6-membered ring unit. So when we talk about a simple 6-membered unit, the paracymene type structure is the only thing that is possible for this kind of terpene. In other words, the gem-dimethyl group is actually kept outside of this ring. So this is the basic gem-Dialkyl rule. And in fact, this gem-Dialkyl rule is actually given by Thorpe and Ingold. And when they talk about this Thorpe-Ingold effect, that is basically a different type of gem-Dialkyl rule, where they say the rate enhancement due to the intermolecular cyclization, that means small and medium size ring, they talk about. In that particular case, what they say is the gem-Dialkyl rule actually favors the quick and the easy formation of a 6-membered ring, especially the small and the medium sized ring are formed very quickly because of this gem-Dialkyl rule. And on the other hand, in the case of terpenes, especially for a monocyclic 6-membered ring, say when they talked about the small and medium sized ring, they talked about 3-membered, 4-membered, 5-membered and 7 and 8-membered rings separately. So the small one refers to 3, 4, 5 and the medium refers to 7 and 8. They did not talk about 6-membered ring. They talked 6-membered rings separately. So the rate enhancement is one type of rule. Another, stability, is a different type of rule, but both are proposed by Thorpe and Ingold. So you have to be very careful when we are talking about terpenes, especially the 6-membered ring. Those 6-membered rings having the gem-Dialkyl unit and the double bond within the 6-membered ring is unstable. But again, there are some exceptions. Let us look at the exceptions. Let us look at another case when we talk about the bicyclic monoterpenes. Here we have a 6-membered ring with either 3, 4 or 5-membered ring. And when we have these kind of systems, the gem-dimethyl group actually stabilizes this ring. So there is a major difference. When we talk about the monocyclic system, then the gem-dimethyl and the double bond destabilize the system, whereas when we talk about the bicyclic ring, actually this presence of a gem-dimethyl group here or here and here. So we actually have a 3-membered ring, we have a 4-membered ring, we have a 5-membered ring in combination with the 6-membered ring. So that is where we have 2 or the bicyclic systems. The presence of gem-dimethyl group actually stabilizes these kind of compounds, and we actually have examples of various these kind of monoterpenes. Bicyclic monoterpenes are actually known and they are quite stable. So here when we talk about the monocyclic system of sesquiterpenes, that means when we have 3 terpene units that are present in these kind of compounds, we have let us say 1, 2 and 3 isoprene units are present here. So in all these cases, the gem-dimethyl group, because we are talking about a monocyclic 6-membered ring here. So when we have a monocyclic 6-membered ring, the gem-dimethyl unit is actually always present outside of the ring. So here again, if you look at the gem-dimethyl is actually kept outside of the ring, and this 6-membered ring does not have the gem-dimethyl unit when it is a monocyclic system. So that is the major difference with respect to the gem-dialkyl rule. If it is a bicyclic system, the gem-dimethyl stabilizes the ring. If it is a monocyclic system, the gem-dimethyl destabilizes the ring. And of course, we will just briefly go through some of the challenges faced during the isolation of terpenes, because as we mentioned earlier, all the terpenes are basically present in aroma plants or essential oils. So in other words, they are actually found as a mixture. So the terpene mixtures are basically isolated by the distillation method. So the essential oil may contain lots of terpene, and each individual terpene can be isolated by distillation. But when we do this distillation, so what happens is, since we have a mixture of terpenes, sometimes high boiling or low boiling terpenes are lost, because they may not distill. The low boiling one may distill even before reaching the distillation temperature, because they are so volatile, we cannot even cool it efficiently to isolate them. So the low boiling terpenes can be easily lost, and the high boiling ones will never distill out because they have the very high boiling point. So in those kind of situations, during the distillation, we may actually lose either the low boiling terpenes or the high boiling terpenes, and in some cases some of the terpenes may even undergo a chemical change, because as we mentioned when we talked about the diterpene formation from the monoterpene, so the isoprene rule, before it was identified, the isoprene unit 2 C5 units combined to give a C10 unit by simple heating. So this is one of the method that was actually used to form the isoprene or the diisopentene unit. So many times what may happen is, during the distillation, since we are heating, some of the terpenes may actually undergo a chemical change. That means entirely a new compound may be formed during the distillation process. And of course, when we talk about the global nature, terpenes are present in various plants. So the plant may be produced either in India or maybe in different parts of the world. So due to various changes, especially the place or the environment at which the plant is actually grown, or the geographical locations, or the soil type in which it was grown, or the age of the plant, or the various different weather conditions, cold, hot, or arid conditions, or the different types of fertilizers used at the different time of harvest, all these things leads to a different composition and nature of the terpenes that is present in a particular essential oil. Of course, what will change mainly is the composition may change, and in some cases even the nature of the terpene may change from place to place. So we have to be very careful while we are isolating these terpenes. And again, some of the degradation that may occur during the storage is like if we keep it for a longer time, some of these terpenes may undergo air oxidation. So because we have the isoprene units or the terpenes have a double bond, so these kind of double bonds can easily undergo oxidation. So that is the reason some chemical change may occur if we store this essential oils for a longer time. And if the conditions are very poor for the storage, that also leads to some degradation in the essential oils. So we have to be very careful while we are trying to isolate terpenes from a mixture. And in fact, when we talk about India, Sukhdev is a very well-known Indian scientist who actually worked in the case of terpenoids. And he had actually discovered a couple of new terpenes like sesqui and diterpenoid compounds. He had identified, that is the new skeletal structures he had actually isolated and characterized it. And he is one of the leading scientists from India who worked on terpene chemistry. And if you look at some of the journals, we talked about the isoprene rule. So the isoprene rule was given by one particular journal article, and similarly the Journal of Endocrinology had talked about lot about the isolation and the occurrence of various plants. If someone is interested, they can actually read these papers for further information. Let us recap in the summary what we have studied so far. We have studied about the occurrence of various terpenes in nature. So what are all the different spices, herbs and other things where the terpenes are present, we have seen. We have also looked at various plants and flowers. They contain terpenes in different forms, we have seen. And we have also studied about the various terpenes that are present in medicinally useful plants. Later on we studied about the isoprene rule, how the isoprene rule was developed, and how the head to tail combination and tail to tail or head and head combination leads to different types of terpenes we have seen. We have also looked at various examples like lanosterol, squalene and other things, how the isoprene rule can be applied to these kind of compounds we have seen. And we have also studied about the gem-Dialkyl rule, like how the gem-Dialkyl rule talks about the instability of a C6 system alone, whereas the same gem-Dialkyl rules talks about the stability of a 6-membered in combination with either 3, 4 or 5-membered ring. So the bicyclic systems are stabilized by the gem-Dialkyl rule, whereas the monocyclic systems are destabilized by the gem-Dialkyl rule. So these are all the things we have studied about terpenes in this particular session. With that, we conclude this session. Thank you.